Okay students, in the last video class we had actually studied a lot about your androecium. We had covered up a lot of things regarding the pollen structure, and even about the basics about a male gametophyte. What exactly is a male gametophyte? What are the nuclei which make it up? And how the tube nucleus plays a very important role in creating the pollen tube, by the help of which the male gametes are transferred into the gynoecium of the other flower in which this pollination has taken place. So after studying a lot about this pollen grains, so let us now focus a bit about a very short part that I need to cover, that is about the dehiscence of anther, which is a very important thing, because until and unless there is dehiscence of anther, we will not be able to transfer the pollen which has matured and which is out of its pollen sac in order to be transferred to the stigma of another gynoecium. So dehiscence of anther means the pollen sacs and the wall of the anther, they have ruptured. Okay, they have ruptured, and since they have ruptured, the pollen are free. And they get transmitted into the surroundings. Now from here the process starts, that how the pollen grains will be transferred to the gynoecium of the flower of the same species. Now it might be of the same flower, gynoecium of the same flower, it might be the gynoecium of another flower. If it is the same flower, we call it as the self-pollination. If it is the flower of the same plant, another flower of the same plant, or another flower of the same species in a different plant, then also we call it as a cross-pollination. So whatever it is, when we are talking about dehiscence of anther, let's stick to this. Pollens are free, no doubt, but how does this happen? How does this entire thing happen? What causes it? How is it that so easily the walls get ruptured? Now the thing is, actually with time, once the pollen grains, they are present inside the pollen sacs, with time they show desiccation, or you can say drying up. Once they start drying up or they lose water, what happens? They were generally completely intact or properly attached to the walls of the pollen sacs with the help of the callose. We had discussed about callose and the callase, the role of callase as an enzyme of digesting the callose, which actually helps in the attachment of the pollen or keeping them in position in the pollen sacs. Once that callase enzyme starts acting on the callose, and simultaneously they lose water. So it's actually a two-step process. One is they lose water. Second is the callase enzyme, it starts acting on the callose, because or by the virtue of which the pollen grains which were actually intact to the pollen sacs or the walls of the pollen sacs, they actually start moving away from it. If I say that this is a pollen sac, they were attached to the walls. After desiccation or after getting dried up completely, the structure looks something like this. They are away from the walls, means they are in a loose state, loosely held state. They are no more intact or attached to each other. Once there is any small jerk also they experience, easily they can move out from the pollen sacs. Now talking about how this rupturing takes place, definitely if the callose content is getting deteriorated and they are starting to lose water, the cells are also going to show some sort of dehydrated state. They are also going to lose water, by the virtue of which the cells of this pollen sac walls, which were actually thick at that time in the beginning, they start becoming thin, and so thin that they rupture very easily. And once they rupture, all the pollen contents, they come out, and this entire mechanism is called dehiscence of anther. Very important part. Until and unless there is dehiscence of anther, naturally it is not possible to take the pollen out of the pollen sac and get another flower pollinated. It is not possible. Yes, there is another mechanism or another process by the help of which we people can actually get it done. And that process of removing the anther or taking out the pollens of the desired variety of the flower and then utilizing it for pollination purpose, it is actually called emasculation. And emasculation is actually performed in order to obtain a desired variety. Whenever you need a desired variety of plant, you need to perform emasculation. You take the pollen of one particular flower of one variety, of course of the same species, and you take it to the other flower for the gynoecium to be getting pollinated, and then the further changes which will take place in order to give you something which you desire for, the desirable complementary characteristics. Now, emasculation, another thing, emasculation is performed only in those flowers in which both the androecium and gynoecium are present. Now, in case of those flowers which are unisexual, which are not hermaphrodites, which are not bisexual, in that case they take a normal course, they take a very natural course in order to perform the pollination or get the anther undergo dehiscence, the pollen sacs come out, the pollen sacs rupture, the pollen grains come out, and they pollinate another gynoecium or the another flower. Otherwise, emasculation is performed artificially. It's a artificial hybridization technique, and thus it gives you a desired variety. Now, in the forthcoming part, we'll be actually covering up the gynoecium structure and especially laying stress on the structure of the embryo sac, the cells present inside, assort a bit information about the development of those cells or the nuclei, and then we would go into the fertilization, post-fertilization changes, and then about the seeds. Okay, so let me start with the structure of gynoecium. Okay, so now talking about the gynoecium of a flower. Now, when we say the gynoecium, it is the female reproductive wall of the flower, and it basically comprises of three major parts, that is stigma, style, and ovary. So, if diagrammatically I have to just show you a brief structure, then it looks something like this. The upper lobed part is the stigma, the neck-like projection is your style, and ultimately it leads into the ovary. Now, actually, when we talk about this gynoecium structure, this ovary actually houses a lot of chambers, and those chambers present inside the ovary, they are actually referred to as the locules. Now, you might ask, now what are the significance of these chambers? Is it for someone to reside? Oh yes, it is for someone to reside. And what are those structures? They are actually ovules, and the ovules are not placed in the locules or the chambers as such at a random manner. No, there is a process. Nature has its own process. It is the most disciplined thing existing on earth. Nature has its own discipline. So the ovules are also placed in chambers in a properly arranged manner, and each ovule is attached to a soft tissue in the center. Okay, now for example, if I show you the section of the ovary, for example, so you can see I have drawn some chambers over here. I have drawn some compartments. So this is, you can see in the middle there is a soft tissue. If this is the locule, and to here something is connected like this. So these two structures which you can see, they are actually the ovules, no doubt in that. They are the ovules. Then what is this mass of substance or the connective tissue that is present, the tissue which is present over here, the parenchymatous tissue I should say, to be very specific? It is actually nothing but called placenta. So basically, if anything is asked regarding placenta, what is placenta? You definitely must have heard about the placenta in the human reproductive system, or you can say the reproductive system in which the umbilical cord gets extended, elongated from there, and it connects the womb or the body of the mother to the fetus which is developing inside. So exactly the same performance is shown by the placenta over here. They keep the ovules in place and they provide nutrition, no doubt in that. And the process in which the ovules are connected to the placenta and the manner in which they are organized or they are arranged, that is called placentation. Okay, that is called placentation. Placenta, placentation. The arrangement of ovules in the locules or the chambers of the ovary is called placentation. Now, talking about the placentation, if we see that the ovule is actually attached to the placenta, then there is a point of attachment. So that point of attachment or the structure through which it is attached is called funiculus. So funiculus is the stalk-like structure which helps in attachment of the ovule to the placenta. And then we say that if that funiculus is the part or the stalk that extends out of the placenta, I repeat again, funiculus is the stalk that extends out of the placenta for the attachment of the ovule to it, for the placentation of the ovule, then what is the point on the ovule to which the funiculus is attached, or to which the funiculus has been strongly embedded into? That point where it is strongly attached to the funiculus in the ovule is called I suspect my handwriting might not be legible, so let me rewrite it. It is the hilum. And many a times, actually, you might have gone through this thing that ovules give rise to seeds. Ovules give rise to seeds. And while studying the structure, while covering the structure of the seed in the next few lessons, video lessons, we might be talking about this structure also, the hilum of the seed. So hilum is that particular point of attachment of the ovule to the ovary through the funiculus. So the statement is very important. If necessary, you can pause, rewind, and again listen to it so that you get the exact statement of defining what is or the definition of the hilum. So hilum is that point on the ovule to which or through which the ovule is attached to the placenta through the funiculus. So we have to be actually very specific when it comes about the definitions of the hilum or definitions of anything regarding the structure, any location of a particular structure in biology. Okay, because generally you get a lot of questions regarding the location and function of some organs. So this is one such thing. Now what happens sometimes inside this ovary, when we are studying the structure of the ovary, inside it you find major portion to be filled with mass laden tissue, highly nutritive tissue will be there inside, and only a small part will be there, or the chamber where actually the ovule is rested or the ovule is placed. That is actually referred to as your embryo sac. We refer it as the embryo sac, while this part, this nutritive tissue laden mass of cells, or I should say the nutritive tissue which is present apart from the entire embryo sac, that is referred to as the nucellus. So if I ask that what is nucellus, or if you ask me what is nucellus, the nucellus is the food laden mass of cells or the most nutritive tissue in the ovary which provides nutrition or nourishment to the growing ovules inside. Now talking about that, if there is an ovule, we can see it inside the embryo sac, then what is there any protection, any mechanism of protection for this embryo sac? Yes, there is a mechanism of protection for the embryo sac, and those two layers of which provide protection to the ovules or this embryo sac, they are referred to as the integuments. Okay, they are referred to as the integuments, and these integuments, they are actually two in number. Okay, they are two in number, the outermost and the innermost, and actually their function is to provide protection, no doubt in that. They gain their importance, much more importance, only when this ovule becomes a seed, which is a post-fertilization mechanism. Okay, so right now for this time being, let's only concentrate on the integuments, not the post-fertilization changes. Integuments, they are two in number, outer and the inner integument, and what is the function? The function is to provide protection to the growing ovule inside the ovary. Now talking about if there is a point through which there might be some access to the embryo sac in the ovary. Obviously the embryo sac is just closely, very nicely it has been layered and is surrounded by the integuments. Then further you have this nucellus, that is the protective tissue. If you are asked that is there any point through which one can have access into the embryo sac, then what is that called? So that point is actually referred to as the micropyle. It is referred to as the micropyle, and the end just opposite to the micropyle, if this is, for example, I am saying if this is the micropylar end, then the point just opposite to it in the embryo sac is referred to as the callus. Okay, it is referred to as the callus, and we generally use the term like micropylar end and the callosal end. It is for reference. Okay, for reference we use this term micropylar end and the callosal end. So I hope this basic thing is clear to you. Now, when we talk about that, what is this ovule? What does this ovule contain of? That is again a very important question, that what does this ovule contain of? So let's focus on this thing.